Hello ladies and gentlemen and welcome back to C.A.R. Entertainment. I'm your host Dylan Hamilton, and today guys we're moving on from our gene technology revision videos, and we're now going to do population genetics, starting off with monohybrid inheritance under 822 biology. The odds of you being asked to do a monohybrid in your exam is very unlikely unless it's going to be a lethal ALLELIC combination monohybrid cross, but we're going to go through it anyway because it's a good base to get a good understanding of why and how you build up to a dihybrid. So you do monohybrid and then dihybrid. You're probably going to be asked to do a dihybrid, but we're going to cover monohybrid inheritance as well, just so you have that underlying understanding, and therefore it means the dihybrid stuff's a lot easier. So we'll start off with definitions. So an allele is an alternative form of the same gene. A locus is the position on a chromosome where the gene is, and alleles will occupy the same position on a pair of homologous chromosomes. If you're homozygous, then both your alleles on the homologous chromosome are the same. If you're heterozygous, two alleles are different. A genotype is two alleles for a particular characteristic. A phenotype is then the feature that results from the expression of your gene, which is influenced by environmental factors. You can have dominant alleles, which are always expressed with a capital in your genotype. I should say genotype, not phenotype. And recessive alleles are only expressed if both alleles are recessive. Okay, and these are lowercase letters. So you'll go through and we'll talk about our first cross here, right? So monohybrid inheritance is the inheritance of one character rather than two characters. So you're only gaining one actual characteristic from it. So as each chromosome possesses an allele in a diploid organism, two alleles where each characteristic is in each cell. So let's go through plants, for example, where you can be round or wrinkled. The plant will be either round or it will be wrinkled. A capital R for round, a small r for wrinkled. So as we see, the genotypes we are crossing here is homozygous dominant, because homozygous, two big R's, okay, dominant because the capital R is the dominant allele. Then we have wrinkled, which is homozygous recessive. Homozygous because they're both small r's, and recessive because they're the small r's. Okay, so we cross these by going R, 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 R. You do this through using gametes. Okay, so gametes are like, you see the way the R's are separated. So for example, if we take this left hand side of the R in the blue, where the capitals are, those are split by a little line. So you have R, then a line, then another R. Those are different gametes, for example, and it's similar with the R's on the top. I should state that that big R on the top should be a small r. It's just whenever I try to type it in, it would not correct itself. I couldn't do anything otherwise. I do apologize for that. When we cross that, you get big R, small r, big R, small r, big R, small r, big R, small r. So you get four big R, small r's, meaning the F1 genotype is all RR, which means it's all round because the dominant expresses over the recessive. So let's do our second cross then. So we will take what we've just had there with big R, small r, and we will cross it, and we will cross those. So we will cross big R and small r. So two F1 offspring are crossed to obtain an F2 generation. You usually get a 3-to-1 ratio when two heterozygous are crossed. Okay, so two alleles will separate due to meiosis, and one will go into each gamete, and this is called the segregation of factors. So as you see, big R, small r, we cross these, you get two big R's, a big R, small r, big R, small r, and two small r's. So we have one homozygous dominant, two heterozygous, and one homozygous recessive, meaning they have the phenotypes. We have 3 round and 1 wrinkled. So what's a back cross? A back cross is a way of testing to see what the characteristic is, whether it has a round or a wrinkle. Okay, so if we use the round sided F2, so we take these here. We don't know what. It could be any of these 3 here. So we don't know which one the plant's actually going to have. This one plant, we don't know which one it's going to have without it actually being developed yet. We don't know. It could be either homozygous dominant, heterozygous, or homozygous recessive. So we do a back cross to find out. So we always cross with a homozygous recessive. Every single time. Okay? So we want to determine if it's going to be round or wrinkled. So what we're going to do is we're going to go with a heterozygous round. So we're going to cross a heterozygous and a homozygous recessive, and that will produce a big R, small r, a big R, small r, a small r, small r, and a small r, small r, meaning that we're going to have 1-to-1, 1 round and 1 wrinkled. So what's Mendel's first law and what is codominance. So Mendel's first law, is the man who basically came up with all this here, is that 2 alleles of each gene will separate during meiosis, but only one will enter each gamete. And this is the law of segregation of factors. This is a pretty basic phrase. It just means each gene contains 2 alleles and they'll separate during meiosis, one enters each gamete. Pretty simple stuff there. So what's codominance? Well, codominance is when 2 alleles of a particular gene locus will be different. So you might have a dominant H and a dominant R. Neither of these are dominant or masking over the other, which means the H will not be dominant, will not express itself over the R, and the R will not express itself over the H. Both are dominant. They're none mask the other's effect. So with the absence of these being dominants, essentially you can say they're codominant, so both are dominant or both are neither dominant. The heterozygous phenotype will be different to both homozygous phenotypes. So we will usually get a 1:2 to 1 produced with 2 heterozygote parents crossed for a trait that displays codominance. So the best way we can do this is through codominance crosses. So let's take flowers for an example. Let's say we cross 2 pink phenotypes, okay? So the genotype for a pink flower is C.R. C.W. and we're going to cross that. So in gametes we have C.R. C.W. C.R. C.W. So C.R. 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 will give you C.R. 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 And then you do C.R. and C.W. you'll get C.R. C.W. You'll get C.R. C.W. and then C.W. C.W. So what you eventually get then is 1 C.R. C.R., 2 C.R. C.W. and 1 C.W. C.W. Okay? Now this then gives you 25% of red, because the R, it means red. So you're definitely going to have a red there, because obviously you're possessing the red allele. Then you get 25% are white, because you're only possessing that white allele. But then 50% are pink, because 50% contains the red allele and the white allele. And when you mix red with white, you get pink. And that's where that comes from. So lethal ALLELIC combinations, and these are the sort of crosses you'll probably do in your exam after doing a dihybrid. So this is when multiple alleles have many genes that also have more than 2 alleles, although only 2 can occupy the gene locus on a pair of homologous chromosomes at any one time. So many genes, more than 2 alleles, when only 2 can actually occupy the gene locus at any one time. So what happens then? Well, a lethal ALLELIC combination will occur, and this affects these effects of lethal combination of alleles are seen in the inheritance of coat color on mice. So this is a good example of coat color on mice. Well, yellow and grey coloured fur as an example. The allele yellow is dominant, so the allele for yellow fur is dominant over grey. When you crossbreed 2 heterozygous yellow mice, you'll get 3 yellow, 1 grey, but you will actually have 2 yellow and 1 grey, because when you have 2 alleles that code for yellow, you'll get a yellow combination and the offspring will fail to develop proper embryos. So this is what happens. Whenever you cross it, for an example, you are going to, let's say Y for yellow, G for grey. You will get a YY, you will get a YG, a YG, and a GG. Okay? So what you're going to get there is 2 yellow because you've got the YGs, 2 YGs. You're going to get a grey because you have 2 Gs. But that one that's 2 Ys, that offspring will fail to develop a proper embryo, and that's a lethal ALLELIC combination, and therefore that won't actually be produced. So instead of 3:1, it's actually 2:1. The exceptions is that in non-lethal combinations that you will survive long enough to be counted in the progeny. A lethal ALLELIC combination is most often going to be recessive, with only being expressed when both alleles are present. And a good exception is the allele for Huntington's disease, which is inherited in dominant autosomal lethal allele, because that's what it is, it's a dominant autosomal lethal allele, which is presence always leads to death, which is unfortunate. But the difference of all our lethal allele combinations that Huntington's disease has is that its effect will only manifest themselves in the adult without prior warning during childhood. So you actually develop your embryo, you grow old with this allele, this lethal allele condition, but you will die when you're about 40 or 50. So at least you get some form of life out of it. Thank you for watching this video. I've been Donald Hamilton from C.A.R Entertainment, and I will see you next time.